So now we're going to get into this major group of animals called the annelids. All right, and these are referred to as the segmented worms. So I'll talk specifically about the segments and how that is sort of broken up as we kind of go along. This is going to be triploblastic group as they develop. So the three true tissues, endoderm, ectoderm, mesoderm, they're going to be now true coelomates. So that means if this is here a cross section here, kind of right there, cross section, we'll have a body cavity. So that's going to be this cavity here, which is going to be the coelom. And in this case, it's a true coelom because the cavity is lined on the outside with mesodermal tissue, and then to the inside with mesodermal tissue. So the digestive cavity or gut, so this will be the intestine here. It itself, the tube, is made up of the endoderm. That's where it's derived from gastrulation. The tube would grow toward the interior, differentiate to become new tissue, but then it'll be surrounded on the outside by mesodermal tissue. So that cavity is a true coelom, and then it's going to give rise to a variety of new structures that we're going to talk about with this group. Okay. This group will display a characteristic called metamerism, which we'll get into, and that is really the repetition of structure. And what we'll find is that this has to do with the segmentation. So as the body is broken up into segments, we will see repetition of structures within those segments. Everything is not completely metamerized, as if there is going to be a digestive tract, a tube running right through the middle of it, and that's just one continuous tube. It's not like duplicated or replicated every single time. So this is different than in the cestode flatworms. So in the platyhelminthes, the body is a whole bunch of these segments, which we refer to as proglottids. But those are not actual segments in this particular sense, in that there's no direct connection all the way through, all of them. Each one is its own sort of almost independent, asexually produced, unique structure. Whereas these are all part of one organism, and then there's just a repetition. Later we're going to get into, not in this lecture, but soon we're going to getting to the point where we'll talk about genes that regulate development in animals, genes called Hox genes. And as we get to those, we'll look at how these genes control development of structures within segments of animals' bodies and how they can be then duplicated or expressed multiple times to make copies of the same sort of structures. So we'll kind of get into that. Before we get into just listing all the terms there, we'll get to some of those. I'll talk about now some of the overall structure of the animal. So it has a digestive system. We said the complete digestive system through it. So this would be the mouth, up here. And it's a complete two openings. Everything is going to move all the way through. Now they're going to have differentiation within the digestive system. So we're going to kind of use this as a basic model to kind of build the rest of it, and I'll add into it as I draw along here. So after the mouth, we have a muscular structure. So it's not, instead of just a tube, just a non-muscular tube. Now, because we have additional mesoderm, we have a muscular tube that has much more complexity to it than, say, we had in the nematodes. This is our pharynx. All right, and the pharynx is going to be muscular and help us kind of pull food then into the mouth. Now, some groups of annelids, we'll talk about the polychaetes, which are marine worms. They will actually have a jaw-type structure that they can use to bite or pierce into their prey. The leeches have a unique type of, leeches or annelids, they're going to have a unique type of structure with this rasping that they can use to damage tissue. But the earthworms, the oligochaetes, which we'll talk about, they don't have a structure like that. They're just kind of pulling stuff into the mouth. So we have a pharynx. Behind the pharynx is a tube, is the esophagus. Then, coming next, is a structure called the crop, which is more of a storage structure. because now food slows down a little bit as it goes into this next section here, which is called the gizzard. It's very muscular. In addition to that, it often has little calcified plates inside it that it can use for grinding. So the gizzard is a grinding structure. So essentially food is brought into the mouth, taken through the esophagus, stored in the crop, and then little by little it's pushed into the gizzard. And then as it goes into the gizzard, it's ground up into very, very, very fine material before it passes then into the intestines, where it'll just continue and nutrients will be kind of absorbed out of that material as it moves along. And then eventually whatever isn't absorbed just becomes waste and is then removed from the animal at the posterior end. So we have the anterior end where the mouth is, and the posterior end where the anus is, and then the waste will come out there. And so we have this differentiation in the digestive system. Now the reason I'm kind of pointing this out first, we're going to use it to build off and talk about some of the other systems that this group has. One of the next things they have that's going to be now unique, totally unique, is that they're going to have a circulatory system, not unique among animals, but unique in what we've talked about so far. No animal group so far that we've talked about has had a circulatory system. So here we have a series of tubes running through the animal's body, and in addition to that, we have blood, right? So they do have blood with hemoglobin. It is, however, free hemoglobin, so our hemoglobin is in red blood cells. The red blood cells then have the hemoglobin, which then binds oxygen and carries it through our circulatory system. They have a circulatory system with blood vessels, and inside those blood vessels are fluid, plasma type fluid, but then there's just hemoglobin in the blood. Right. What they have are structures surrounding the esophagus. So a bunch of ring-like structures. And I'm not drawing all of them. This isn't the full detail. Typically they line up with segments of the animal's body. Again, I'm not getting into all the detail. But these rings are often referred to as aortic arches. And another term for them are pseudo hearts, because they're these tubes that wrap around the digestive tract, and then they contract, and what they'll do is they'll pump and push blood. Now the animal's going to have running to the anterior, right along the top. If you do the dissection of this, you'd be able to see this. It's pretty obvious structure. There's going to be a back or dorsal blood vessel, and that would be kind of represented here. This would be the dorsal blood vessel. And then obviously this would be the ventral blood vessel. And which could be kind of hard for me to draw in here because it would be the opposite side of this screen, but there'll essentially be another blood vessel running along the underside of this structure. So along the top or the dorsal side of the digestive tract is a blood vessel along the ventral side. And then there are these rings, these aortic arches, that connect them together. In addition to that, all along the animal's body, there are going to be connections between the dorsal and the ventral blood vessel. But these happen to be very, very thick. Generally what happens is that the blood is kind of pushed along the dorsal blood vessel from posterior to anterior. So blood generally flows this direction on the dorsal side, and then it's coming back along the ventral side. However, the blood flow is a little bit, it's not like our circulatory system where it's very directional and then there's valves that like shut off so that we can't have backflow. There are no valves there, so there's a lot of backflow. So the moves and then it kind of moves back, and then it moves and it sort of moves back. So it's not exactly the way our system is. It's its own sort of primitive system, but it's the beginning of types of circulatory systems we see then in other animals. Now, related to that is something we started talking about a little bit before with the flatworms. They had structures called flame cells, which we also refer to as the protonephridia, essentially as a way of filtering out waste material and regulating osmosis in those organisms. And we say they kind of then resemble the beginnings of structures that would be kidney-like. Now we start to continue that along and get something even a little more complicated. So we have nephridia. So this structure here, this kind of squiggly tube that I drew in a segment, so it's going to be a segment of the animal's body. And if we start to actually, look at the body itself, what's going to happen is the body wall of the worm going to go like this. What we're going to find is that there are going to be septa that are going to divide the animal, right, into segments. Like this. That's why we call them the segmented worms. Again, I'll talk a little bit more about that in a bit. And so that's kind of what we're looking at in this picture here. We're looking at, say, like one of these particular segments. And so in the segment, what you're going to have is the nephridia. So what the nephridia will do is they'll have an opening here in the coelom. So this is the coelomic compartment. And they'll be able to take in fluid from the coelom and then pass it through these series of tubes that are going to be acting as filters. And then waste material that is collected will be then pushed out of the animal's body through pores in the body wall. So waste will actually just come out all along. So in each of the segments, there'll be a place that this can happen, right, on both sides of the animal's body. So that's all waste kind of being removed. Now what you can see here, this is again representing the dorsal blood vessel, ventral blood vessel, and you can see these little squigglies that are pink here, kind of the same color, are going to be capillaries. So that's significant in that, say, our kidneys, which are filters, are heavily integrated with the circulatory system. So there's a lot of blood vessels associated with the filtration structures, so it's kind of filtering the waste then out of our blood and then collecting it, and then also conserving water and doing all those osmoregulatory functions, as well. So that's what we kind of see here. We see, in addition to just taking the fluid of the coelomic cavity and filtering it, there's an association with the circulatory system. So waste from the blood can be pulled out of the organism and then also collected right here in these nephridia. So that's another step forward in the development of a filtration system for waste. As we get an animal that's more and more complicated, what we'll see is that the more simple animals can have a lot of exchange of gases across the body wall, both in and out. So waste gases out, oxygen in. Nutrients can just be absorbed. Waste can actually be diffused out. Diffusion across the body wall can happen in organisms that are very simple and have very thin, non-complex body walls. But as you start to get more and more complex, as you get more layers of tissues, you have thicker body walls, there starts to be more waste and accumulation of material inside the animal that has to get out, and it's not going to just happen by diffusion. So now we need transport systems within the organism to collect that waste and move it out. We're also going to need systems to bring nutrients and gases and oxygen into those other tissues and organs that aren't exposed to the surface, that aren't going to easily be able to exchange gases through diffusion. So outer layers across the cuticle, yes, easily they themselves can get oxygen. But what you're then going to need is have blood vessels pick up that oxygen and then start to carry it through to the other tissues that aren't going to receive oxygen just through simple diffusion. Okay, so you kind of have a combination going on there. There's a little bit of diffusion of the oxygen across the body wall, but then you have blood vessels picking up the oxygen and then transporting it. In addition, you have the blood vessels picking up wastes, taking it to the nephridia and then filtering waste out and carrying it out of the animal. So we kind of start to build more organ systems as we get into this group. Looking at the body wall itself, let's kind of break that down. They will have a very thin, non-living cuticle. And it's easy gas exchange across the cuticle. Now, there's pressure that can build up inside the body cavity, but the body wall has double layer of muscle. So we're actually going to have circular So actually, well, beneath the cuticles, the epidermis, and the epidermis secretes the cuticle. So the cuticle is the outermost layer. Then we have the epidermis. Then we're going to have circular muscle next. And then we'll have longitudinal muscle following that. So we have two muscle layers, if you remember now back into the nematodes. We said the nematodes only had longitudinal muscle. They lacked circular muscle. They didn't have it. But this group has both circular muscle running along the animal's body and then longitudinal muscle. Now, they also will have these mesenteries that will kind of divide the body up into segments. The significance of this, if holding this segment here, is that the musculature can then contract and affect a single segment. So that means one segment of the animal's body can have the circular muscle contract, so it kind of can squeeze that tighter. That doesn't affect this segment or this segment. These two segments remain independent in terms of that muscle contraction. So the body then has much more control over its ability to move, because it can have a much more fine control in each individual segment, or whether it's kind of squeezing that segment thinner and elongating it, or the longitudinal muscle activating and then contracting the segment, making it thicker but shorter. And then that movement carried throughout the animal's body allows them to move. Right? So they can kind of then burrow their way through tissues and sediments, and then we are going to talk about in the polychaetes, when we get to that group, they'll have other ways of locomoting. They'll be able to move with little appendages, so little paddle-like appendages they can use to swim through the water column. Something I didn't bring up yet or talk about, and it's kind of an important feature for this group, right, is that the body, now sticking out of the cuticle like this, in earthworms, the annelids, it's something we don't really see that much. They're highly reduced. But there are sort of hair-like structures that stick out of the animal's body, right, like this. So in earthworms, this is something that's also going to help them as they burrow through sediment. So as they move forward, pushing themselves through sediment, when they start to contract in the other direction, they could just slide backwards, so going forward, backward. But what happens is these little hair-like structures, these little setae, kind of extend outward and act like little anchors. So if you have a worm like this, these little setae will then stick out like that. So as the movement would sort of normally be backward, they act as an anchor and hold them in place. So then they can pull the rest of their body forward, and then they'll push this through the sediment, and as they do, as they kind of extend this segment, the setae lie down flat along the animal's body so that they don't cause any resistance. Then when the animal goes to pull its back end forward, those setae will collapse, but these setae will stick out like little anchors, and they can kind of alternate this throughout the body, so they can kind of push and pull themselves and anchor themselves and move in a particular direction. Some of the other organisms, again, the polychaetes, so setae in this term, chaeta, these are the same. You're going to find this unfortunate thing happening a lot as we study biology in general, especially animal biology and structures. There are many times where there's the exact same structure that just has multiple names. And so setae and chaetae, C-H-A-E, are the same exact thing. And they're just these little hair-like structures sticking off the cuticle of the animal's body that they can control and stick out or pull back in. The polychaetes, many, are going to have a lot of these, so their body's going to be covered in them. Some of them will be, so they're called like bristle worms because they're so covered in them. And they can be defensive and have a variety of other characteristics as well. So, as far as systems go, so we have talked about the muscular arrangement, the body wall cavities, they do have a circulatory system with hemoglobin. They have an excretory system. I didn't mention the nervous system really here. There's not much new, except we just have a little more well-developed ganglia. Okay, so we have two ganglia here and then a ring. So that goes right here or in the front, and then it leads to a ventral. So it's going to be a ventral side nerve cord. Ventral, and that's the nerve cord. But they're going to have this ring, this anterior nerve ring connected to these two ganglia. So it's kind of like the brain of the animal, but again, they don't really have a true brain yet. They have these separate ganglia. And then that will have little branches of nerves that go to each segment that interact with the muscle that allows them to coordinate their movement. So again, there's a lot to really go into. That could spend a very long time on this and multiple lectures, but we're just trying to introduce the basics of the annelids, their overall body wall structure, there being a triploblastic coelomate, having septa dividing the body into different segments. However, while those segments have certain things like the nephridia and certain capillaries and other structures that get repeated, there are other things like the circulatory system that are unique. So only up in the anterior region do we see these aortic arches. We don't see them all throughout the body. We see the circulatory system, we see a blood vessel running all the way through continuously through the body, through the dorsal side and the ventral side. We then have differentiation in the digestive tract. So a muscular pharynx up front, right at the anterior region, then a thin esophagus, a crop for storage, and a gizzard for grinding. And then the rest of it is all intestine, just absorb nutrients and exchange and conserve water and all those sorts of things. So the next thing is I'm going to continue with annelids, but we'll look at the classes of annelids, and I'll kind of point out a little bit more about what makes some of the classes unique and some of the details they have. We'll kind of add to a little bit of this as we go into the next lecture.